Thank you so much. And it really is a pleasure to be with you. I'm sorry that it's remote. It's never quite the same as in person, but I hope to be able to join you on a future occasion. Um, I think what I'd like to try to do over the next, I don't know, half hour that I have is talk to you a little bit about our thoughts about ALS prevention. And I want to talk to you a little bit about the past, where we began the journey and what the journey has looked like to get to the present day of now running the first pre-symptomatic clinical trial, and then to share some thoughts about where we think that can lead us in terms of the future as we try to get beyond the subset of people with SOD1 who are in our ongoing pre-symptomatic clinical trial, and how do we broaden this to other genetic populations and potentially to people who are at risk even without genetic causes. And I'm hoping, Martin, that I'll leave enough time for us to have a dialogue and that we'll be able to do that despite the remote nature of this. So here are my disclosures. It's important I spend a moment here. I have served as a site investigator in industry-sponsored studies, most recently by Biogen and Orphazyme. I do some consulting, mostly around helping industry design clinical trials, both in the ALS and the myasthenia space. I've listed my research funding there. And then importantly, a provisional patent. And what's not here, but should be, is that the University of Miami has licensed some of our data to Biogen to help with the design of one of the clinical trials that I will talk about. So what I want to do to start is to go back to 2006, which is roughly when this began for me. And I pulled up one of the early grants that I submitted to the Muscular Dystrophy Association. And this was actually a tough sell back then. And somebody talked me out of calling this the PAPLS study, the Predict and Prevent ALS study. But I highlight this because we were very much interested in both prediction, when people at genetic risk for disease might develop disease, and how do we gather the information that we need in order to prevent disease. And so this was some text from our abstract that we said new information regarding genetic risk combined with disappointing results from clinical trials has prompted a shift in attention towards the goal of prediction and prevention. And we said that families with ALS due to mutations in SOD1 represent a population of high-risk individuals who can be targeted in the clinical trial designed to delay or prevent the onset of ALS. But in order to pursue the goal of a prevention trial, it's crucial that we better understand the basic biology and epidemiology of SOD1 ALS, and that we develop methods for recruiting, retaining, and studying this population. And I think that's still true. The only things I would change is to say that we've now learned much more about the genetic basis for ALS, and this goes well beyond SOD1, and you could substitute any SC9 or any other or all other genetic causes, and I think the same would be true. And so, as we started down this road, we were interested in studying pre-symptomatic disease with a view to prevention. And I'll try to show you that as we've tried to accumulate bits of information that we needed to be able to do this. So when we set out, we identified a number of challenges. One was whether it would be possible to identify, recruit, and study a population of people at genetic risk. And it's hard to imagine that today, but back in 2005, 2006, even before, that wasn't clear. We didn't know what the interest and willingness would be of potential study participants. In fact, I remember early grant reviews where we were told by reviewers that nobody would be interested. How would we do genetic testing and disclose results? We had something to learn from other disease communities like the Huntington's world, but this really hadn't been done much in the ALS community, and there was some resistance and reservations to this. We were concerned, as were other communities like the Huntington's communities, about the potential psychological harms of learning that you're at risk for a fatal neurodegenerative disease, and how do we mitigate those harms and make sure that we're looking after people in the course of communicating that information. And then we had to navigate a bunch of logistical, legal, and ethical considerations around this whole endeavor. In addition to those issues, we did not know anything about the phenoconversion rate. And I don't know if that term has come up today. I'm sorry that I haven't been able to join for the whole day. Phenoconversion for us means people who develop clinically manifest ALS or FTD from a pre-symptomatic state. And we didn't know what was the annual rate with which people who were carriers would develop clinically manifest in the SOD1 population ALS. And this is obviously a critical piece of information that we would need if we were going to think about the logistics and the feasibility of a preventative clinical trial. At that time, we had no biomarkers. And not only did we not know the phenoconversion rate, we really didn't have a definition of phenoconversion. How would we operationalize this? We had no clear choice of therapeutic candidates, but we did know, depending on what those therapeutic candidates were, the timing of when we could reasonably initiate that therapy might very much depend on its potential side effects and toxicity and what would be the potential long-term safety considerations. And so this is really the blank state of affairs, I think, in the ALS world when we began. So this was one of our early publications, and this was really based off of studying several hundred familial ALS pedigrees. And this paper was entitled "Preventing Familial ALS, Is a Clinical Trial Feasible?" And we were beginning to grapple with how we might think about this. And so we sort of looked at age-adjusted 5-year risk of developing ALS. And we picked 5 years because we thought maybe we could do a clinical trial where we would enroll people and follow them for 5 years. And what you can see is that the 5-year risk of developing disease is actually quite low. There's a group here in the sort of, 50 to 60 range where we're in the mid-teens or getting up to 20% range that might be able to power a study. But as we thought about this across the spectrum, even if we had a drug that could delay the time to onset of disease by 50%, and with 80% power, we like 90% power in a clinical trial, we're looking at a sample size of close to 1,000 people. And the reason that clinical trial would have to be so big and be underpowered is because the 5-year risk of developing ALS was actually quite low. So that was one thing that was clear was a challenge we had to overcome. The other thing early on is we wanted to understand people's attitudes toward genetic testing and interest in a clinical trial. I've drawn your attention here to the SOD1 population. That's what we were studying at the time. But we asked people, this is several hundred, would you be willing to undergo genetic testing to determine whether you're at risk? And overwhelmingly, people said yes. Would they want to know the results of genetic testing? The group was more mixed. And would you be willing to participate in a clinical trial designed to delay the onset of disease? And the community was very interested. And so this empowered us to move forward. So we knew there was an interest and a willingness, and we preliminarily thought that the phenoconversion rate was low. And we recognized that identifying biomarkers that might help us predict who is at greatest risk for phenoconversion would be critical, because that would enable us to enrich a population, enroll people who have that biomarker that tells us they're at the greatest short-term risk, and whom it would then be possible to do a clinical trial. So this study of families then led us to initiate the PRE-FAL study, which we started in 2007. These sorts of studies are now more common, but this was a longitudinal and natural history and biomarker study of people at genetic risk for ALS. And it includes carriers of any ALS-causing genetic mutation. And at the time, this was the only population that we knew to be at an elevated risk for ALS. And although we started with SOD1, this has grown as we discover as a community other genetic causes of disease. We've grown that eligibility. And importantly, we've always felt that we needed to be broadly inclusive, that we wanted the community to be able to participate in and contribute to this research endeavor, even if they did not wish to learn the results of genetic testing. So we built that option into the design of the study. The goals were to identify biomarkers of pre-symptomatic disease, and importantly, biomarkers that could predict when symptomatic disease is coming, when phenoconversion is coming, and to help us prepare for a prevention trial. I just want to underscore that although pre in PRE-FAL stands for pre-symptomatic, we also think about it in terms of prediction, preparing, and ultimately preventing. So a very useful prefix. So just to give you a little bit of a feel for what the study looked like, and still looks like in many ways, we would identify familial pedigrees, we would identify the genetic cause of disease, we would identify unaffected family members. If they'd already undergone testing and knew they were gene negative, we did not enroll them. If they knew they were gene positive, we did, and we would follow them. And if they didn't know, then they could decide, did they want disclosure of their genetic result? And if they did, did they want disclosure? And if they did, they would get genetic counseling, and only the gene positives would be enrolled. But if they did not want to learn their results, then we would enroll both gene positives and gene negatives, and all of the investigators except our statistician would be blind to this. And this was important, enabled us to be broadly inclusive, but also meant that we would bake into the design of the study gene negative or non-carriers from the same families that would, one, keep us blind and masked to our evaluations, and so keep us honest, so to speak, and objective, but also potentially provide a useful control group as we were trying to develop these biomarkers. Initially when we started out, genetic counseling, both pre-test and post-test, the industry standard was to do this in person. And because we were recruiting from around the country in the US, we would fly people in, and both for their pre-test and their post-test. And that put a lot of burden on participants. They were often then away from family. And so early on, we did a small pilot where we randomized people to receive in-person or telephone counseling. And I'll tell you more about that. I recall the genetic counselor I work with thinking I was a little bit nuts. But it's interesting, this has now become, I think, fairly standard that we do a lot of genetic counseling remotely, either by telephone or by remote video link. We were also interested in understanding the impact of testing on people at risk. And so we took a sample of the population and we did qualitative interviews. And we tried to understand a little bit about their attitudes towards learning genetic results. Now that we were offering this, what did we find? Well, actually, more people wanted to learn their results once it became available. And that was actually different to the Huntington's disease community. And we learned something about the potential psychological impact of learning results. And I encourage you to look at the paper if you want to know more about that. But in short, what we learned is that this is obviously a stressful undertaking, but that with the right support systems in place, people would go through a stressful period, but then return at some level to their sort of baseline level of anxiety about knowing that this disease was potentially in the future, but feeling empowered by having learned that information. And as we continued to follow people, we found that actually people were able to adapt to learning about this genetic risk and cope with it quite well. We learned about perceived advantages and disadvantages of in-person versus telephone counseling. And then we defaulted to telephone counseling, although continued to offer people to come in person if that was their preference. And I'm highlighting all of these issues because they've now become standard in what we do, but I'm trying to give you a flavor for all the things we didn't know, because there are many things we still don't know. And I think we have to be meticulous and iterative as we figure this out as we move forward. So we'd understood this interest and willingness. We'd figured out some of the potential psychological harms. We still knew phenoconversion rate was low. Not much had changed there. Along the way, we acquired a lot of experience with genetic counseling and thinking about this in the pre-symptomatic space. And we published some recommendations for how this should be done. One of the things we learned actually from our experience was that in addition to the industry standard of pre-test and post-test, we introduced a third counseling session, what we call pre-decision counseling, where we would discuss with people their motivation for wanting to go through testing, make sure they know they have the freedom to change their mind, explore some of the potential psychological import, their social support system, discuss some of the medico-legal issues, and really give people an additional opportunity to think about whether they wanted to move forward with pre-test and then post-test counseling if they move beyond that. So we ended up being able to address many of these logistical issues. One of our early forays into trying to figure out biomarkers, we had a big interest in potential neuroimaging markers. And this was a study we did looking at magnetic resonance spectroscopy and actually in the spinal cord. And we had a big voxel in the cervical spinal cord. And what you can see here is that we found differences between healthy individuals in blue and patients with ALS in red, and importantly, the pre-symptomatic group sort of spreading across them, suggesting some are more like controls, some are more like ALS. And this was encouraging to us that we could see the signal, but we also realized we were going to need markers that would operate not just at a group level, but that we'd be able to tell at an individual level in you, do you seem to be closer to disease and a greater risk of phenoconversion? Because that sort of marker would really be essential if we were going to move forward into a clinical trial. So PRE-FALS has grown over the years. It's remained really a single center study. It's several hundred in size. It's mostly SOD1, but a good mix of C9 and other genetic causes. But because we've been going for as long as we have, we've accumulated a lot of person years of follow-up, and we've observed a good number of phenoconverters, both to ALS and to FTD, amongst the non-SOD1 population. I think one of the sort of big observations that we made going back now 5 or 6 years was that neurofilament light could be a very useful biomarker, even when measured in blood. And again, I think this has become well known now, but this wasn't at the time. I think you've spoken about neurofilaments today, so I won't belabor this, but these are major structural components of nerve cells that once nerve cells die and degenerate, the neurofilament can be found in the spinal fluid, but importantly, it can be found in blood as well, and that's key. And there are different forms of these. There's light and heavy chains. And I think one of the important things is that we have excellent assays in lab to be able to measure these neurofilaments very well. What we found, I'm looking at serum neurofilament here, and this is CSF neurofilament, and just sort of at baseline. Initially, we didn't see anything in the pre-symptomatic group. The affected population had elevated levels, both in blood and spinal fluid. But when we looked longitudinally, we found that controls were very stable. And if we sort of marked out in gray here, the maximum value that we see in controls, we found the at-risk group, the gene carriers who didn't develop disease, some people at higher levels, but they remained very stable over time. We knew there were elevated levels in the ALS population. And what we found is that neurofilament went up amongst the phenoconverters prior to this time zero, which is the point of phenoconversion. And if I show that to you on a log scale, just because it's easier to see, we don't have to get into the math of that. Here we have the maximum value we see in controls in gray. This is time zero phenoconversion. And you can see all these shaded circles. These are SOD1 carriers. These are people where there's an upward trajectory, but more importantly, their levels are elevated above this normative threshold within the 6 to 12 months prior to phenoconversion. And it was that information that we thought was really going to be helpful in telling us who was going to phenoconvert and was really beginning to empower us to design a clinical trial. We looked at this in a later cohort. We also had, I think this individual was a C9 carrier going back as far as 4 years. We had a FUS carrier right at the upper boundary. And we've begun to acquire additional information across a range of different carriers. We looked at neurofilament heavy as well. We found it had the same pattern, but less helpful, in part because of the greater degree of variation within the healthy control population. This led us to sort of this hypothesis, which I think is still a hypothesis. We don't have enough information on this yet, but we know that in different genetic forms of disease, the symptomatic phase of disease varies. And we believe that it's probably true that the pre-symptomatic phase that precedes people coming over the clinical horizon also varies in duration. And perhaps it's much shorter for people with this very aggressive form of SOD1 ALS, and perhaps a much longer pre-symptomatic period for those who carry genes associated with less aggressive forms of the disease. So to fill in sort of our progress here, we now begin to define phenoconversion rates based on empiric data. I didn't show you this, but we found at least amongst the SOD1A4B carriers within a 2-year period, we had about a 20% phenoconversion rate, irrespective of age. And we now critically had this biomarker, mostly neurofilament light, that we could measure in blood that was predictive of imminent disease and that we could now use for enrichment. And interestingly, when we presented these data at the International Motor Neuron Disease meeting, Biogen and Tim Miller from WashU were presenting the results of their intrathecal SOD1 antisense oligonucleotide, showing these initial phase 1 results. And that led us to initiate discussions with Biogen about using their ASO for SOD1 in the pre-symptomatic population and using neurofilaments as an enrichment marker. So just a word about tofersen in case you haven't covered it. And tofersen is this SOD1 ASO, so antisense oligonucleotide, a form of a gene therapy. So this facilitates degradation of the SOD1 RNA, and so this reduces the level of the SOD1 protein product that we know becomes misfolded and is toxic and causes disease in this population. Tofersen has since been evaluated in a phase 3 trial. And just to show you those data a little bit, these are in people with disease initiated usually about, or on average, about 8 months after symptom onset. And you can see on the ALS functional rating scale that those in red who start tofersen versus placebo, there's a separation. But at the primary endpoint of 28 weeks, there was no clear clinical or statistical benefit. But in the open label phase, where there's an extension of people on drug and now the placebo group have started active drug as well, you can see there's a greater separation here that's both statistically and clinically significant. But this did not meet its primary endpoint. But importantly, there was a reduction of about 30% in the level of the SOD1 protein in CSFs. That's what we expected. The drug was doing its job. And importantly, it was also lowering the level of plasma NFL, neurofilament light. And this is using NFL in a little bit of a different way to how we had been using it in the pre-symptomatic population. It can also be used in this way to show a reduction in neurofilament in response to the drug. And when the placebo group granted drug, we see the same thing happen. And important, I'll come back to this, and I mentioned it, that the median duration of disease at the time of randomization amongst the fast progressors, forgive the typo, was about 8 months. And that may be just too late or relatively late in the course of disease, which is one of the things we've been trying to overcome. So here you can see we're beginning to make progress. Now we have a clear choice of the therapeutic candidate, at least for SOD1 carriers. And we do have a sense of the safety concerns. You know, in the phase 3 trial, although rare, there were some serious adverse events, most notably an inflammatory reaction in the spinal fluid and in the spinal cord. And although this was rare, it's important that we're aware of it because it might impact for how long we think people are able to take this drug and how much they'll be able to tolerate it. And we don't yet know if there's a relationship between duration of therapy and the frequency of this, but it is an adverse event that we need to be aware of. So again, sort of building on this, we hosted a workshop. Dr. Turner was there, Dr. Aurora was there, I think others perhaps were in the room in early 2020. And one of the major focuses of this was to say, how do we operationally define phenoconversion amongst SOD1 carriers for us to be able to incorporate this into a clinical trial? And this was something that we came away from with a decent consensus from the meeting that we felt we were able to do. And so all of these pieces together have enabled us to design and now initiate the ATLAS study. And the ATLAS study is our pre-symptomatic study of this drug tofersen. And the goal is to evaluate whether pre-symptomatic initiation can delay the emergence of clinically manifest ALS. And even if it can't delay, if it even can't prevent it, can we slow the decline in function if it does appear? So if we start early, can we dramatically slow the course? And we will measure this primarily by looking at the proportion of people with the emergence of clinically manifest ALS within 24 months of randomization to receive the drug. So to do this, we had to get into the weeds with neurofilament. So here's the data I showed you before, it's just plotted slightly differently. And we took this information to build a model of how neurofilament changes over time. And we then set a threshold using a particular assay, this assay that Siemens runs, of 44 picograms per milliliter. That's the amount that we would measure in your blood. And with that, people who developed that elevation, they all went on to develop disease. And amongst those who didn't develop the elevation, one of them did. And so these metrics were actually quite good that enabled us to go forward and design the trial. So what does the ATLAS study look like? There are 4 parts to it. In part A, after people are screened and enrolled, part A is a natural history and run-in study. We send phlebotomists out to people's homes. We do a monthly blood draw and we measure their neurofilament levels. If neurofilament goes up above our threshold of 44, and there's also a 10-point increase from baseline, and they're still pre-symptomatic, then they get randomized to drug or placebo in a 1-to-1 ratio. The primary endpoint is the development of clinically manifest ALS. And when and if that happens, people go on to open label drug. So importantly, the latest people are receiving active drug is sort of at time 0 when clinically manifest disease appears, not 8 months in, as occurred in the VALOR study, the phase 3 trial. We recognize that it might happen that people develop disease without a rise in neurofilament. And if that were to happen, they would go straight onto open label drug. So as in part C, the latest they would go on to open label drug is at the point of the emergence of clinically manifest disease. Here are some of the key eligibility criteria. And I'll draw your attention to these specific variants, but to say that the trial is focused on those who carry an SOD1 mutation that is highly penetrant and associated with rapidly progressive disease. And we had to focus on this subset because that was the subset in whom we had good, robust evidence that we could use neurofilament in the way that we have proposed. And I think this is one of the important caveats as we think about getting into other genetic populations. I think neurofilament doesn't behave quite the same way, at least not on its own. And we are almost certainly going to need other biomarkers to help us as we move beyond this SOD1 population. So I've alluded to this, but I think the timing of therapeutic intervention is key. The way I think about disease is there's susceptibility. At some point, disease begins. We don't know when that is or how to quantify that, and people are pre-symptomatic. They then phenoconvert, and there's some lag until they get a diagnosis. And in the phase 3 VALOR study, people were then treated sometime after diagnosis was made. But we know there's a phase of axonal degeneration where the nervous system is sick and neurofilaments are rising here. And so what we're proposing to do in ATLAS, not proposing, are doing, is that we're treating in part B here, when neurofilament goes up. We're also treating in part C and D here at the point of phenoconversion. And what we're hoping to do is to see if this can delay or prevent disease, but also to understand how beneficial this is relative to this and relative to this. And so I think there are multiple things that we're going to learn relevant not just to SOD1 ALS, but I think to all forms of genetic ALS and indeed all forms of ALS about the optimal timing of therapeutic intervention and how we can pull that back in time and engage that earlier. So I'm going to wrap up soon and then hope we can have a brisk dialogue. So I think the roadmap to ALS prevention began with the meticulous study of those at genetic risk. And this was really a partnership with our community of patients and family members at risk for disease or potentially at risk for disease. And the emergent natural history and biomarker data have been critical to the design of the first pre-symptomatic ALS trial in a subset of the SOD1 population. And I want to underscore here, at least in the US, it was important to us that the quality of the data that we provided from PRE-FALS was acceptable to the FDA to help permit the design of the ATLAS study. And you may know this, but tofersen has gotten accelerated approval from the FDA, but it requires additional evidence of efficacy. And ATLAS, if successful, will show that additional evidence of efficacy. And so this is, I guess, a shout out to the importance of this sort of observational data and natural history data that can be so critical to moving forward our ability to get drugs, meaningfully effective drugs, approved by the regulatory agencies. But I think as we think beyond this, I think there are many important lessons, and I hope we'll talk about some of these, that are empowering disease prevention in other populations at genetic risk for disease. So firstly, we've seen an explosion, and I think you've heard about some of this from other speakers today, about cohorts that have grown up in the ALS world, in the FTD world, amongst C9 carriers, amongst other carriers at risk for either ALS or FTD. We need and we're seeing an expanding repertoire of available biomarkers, and neuroimaging is playing an important role here, together with neurofilament and perhaps together with other things that we are still yet to identify. And I think that we do need, we're very likely to need a panel of biomarkers to predict the risk of phenoconversion in genetic populations beyond this subset of the SOD1 population that I think is unique in many ways. And obviously we need folks in the lab and our partnerships with industry to be working and moving forward the development of promising therapeutic approaches for other genetic forms of disease. And I think the idea here is that we don't want to wait until we have those therapeutics and then say, but gosh, we've got 10 years of work we need to do to answer those questions. We want to be developing those tools in those populations in parallel. So the moment we have a therapy that we think, this is the therapy that's actually going to do something that's impacting the underlying biology, that we're ready to go with the biomarkers, the patient population that's interested. We've figured out all of the kinks and logistics, and we can jump very quickly into doing those other disease prevention trials. So before I stop, I want to just acknowledge our study participants and their families. This is a group of people who have been working closely with us and coming to see us every year for now 15, 16 years, and that's an incredible commitment, and we're enormously grateful to them for that. I also want to acknowledge my group at the University of Miami, and especially my colleague Joanne Wu, with whom I co-lead our research group and without whom none of this would happen, as well as my neurologist colleagues, our research coordinators, present and past, Andrea Malaspina, a colleague in the UK with whom we've done lots of the neurofilament work, Dr. Anderson in Sweden, with whom we've done a lot of the genetic work, Christine Stanislaw, our genetic counselor, with whom I started working at Emory and continues to work with us, and then the team at Biogen, who've been great partners in designing and implementing the ATLAS trial, and obviously all of our funding agencies for their generous support over the years. I'm going to stop there. I'm not sure if somebody is unhappy.